much for the opportunity to talk. So today I'm going to talk a little bit about more general title, Systematic Functional Annotation of Large-Scale Biological Networks. So as we all know, people in this room, in the last decade or so, as a field, we have accumulated a huge amount of very large number of biological networks. These networks map all sorts of relationship between genes. And over the years, as we have been accumulating this data, we learned pretty well how to interpret the data we were acquiring based on at the level of individual interactions, at least. We have a very good understanding of what, for example, a physical binding means. We have a pretty good understanding of what a protein-DNA interaction means. We also, in some cases, were figure out how to interpret local structure and modules within networks. We know that if a group of proteins all have physical interactions between them, they're very likely to represent a protein complex. On the other side, however, we have a much worse understanding of what these networks mean biologically on the global scale. What's their global organization? In other words, we actually have trouble understanding what have we learned from mapping all possible interactions of a given type that is not just given by the sum of the individual interactions we have collected. What have we learned from about the system that this network represent? And many of the networks are genome scale, so they do represent the entire system, the entire cell they were derived from. What have we learned from mapping all of them, all of these interactions, rather than a subset of them or each of them individually? So this is a very hard problem, and it's definitely not going to be solved in a year or two. But today I wanted to talk about some of the steps that I'm taking in order to address that question. And as an example for the methods that I've been developing, I'm going to use this network in particular as a test case because I'm very familiar with this network. This network is a genetic interaction similarity network that was produced about 6 years ago. And on this network, every node represents a gene, and two genes are connected by an edge if their genetic interaction profiles are more similar than a chosen threshold. So before I go into explaining how we are how we are looking into this network, I have to explain, at least make sure we're all on the same page in terms of what a genetic interaction is and what does it mean. Why is it important for two genes to have similar genetic interaction profiles? So we have heard a little bit about it earlier today. So genetic interactions represent a phenotypic relationship between genes. And they happen when a double mutant has an unexpected phenotype based on the two singles that you started from. A classical example is synthetic lethality. You have two single mutants that could probably be very, very similar to wild type, but then the double mutant is unexpectedly sick or even lethal. We usually interpret this, many cases, could be explained by a simple parallel pathway model. You have two pathways that impinge on the same essential biological function, such that you can get rid of a member of either one of the two branches, and nothing will happen. But if you create a double mutation, then the function cannot be completed anymore, and the cell is dead. Another example is what some people refer to as epistasis. This is a very confusing term, but all it indicates is that you might have two single mutants that both have some sort of phenotype, and then the double mutant has the same phenotype. This is still unexpected because one way or another you expected the phenotypes of the two single mutants to combine and be, for example, the growth should be worse than either one of them individually. Some of the cases that behave like this can be explained by a linear pathway model. Here you have a single path, linear pathways that impinges on a non-essential function. And when you delete a member of this pathway, you're blocking the entire pathway completely. So adding secondary mutations in the same process will not really make it any worse. Of course, this is an extreme oversimplification of the situation. There is many more cases that don't fit into either one of these two categories. In fact, it's a spectrum of events. And we and others have learned over the years that one easy way to kind of quantify and look at this all is to measure a score for any given double mutant that's given by the difference between the double mutant phenotype and the product of the two singles. The product here is just a convention to indicate what phenotype you would have expected if the two single mutants were independent. There is a bunch of different models that will predict what expectation is. This is just one of them. There is really no systematic analysis to figure out which one is the better, but this one has been working pretty well. And based on this simple approach, you can categorize these interactions as negative because the double mutant is much lower. The phenotype of the double mutant is much lower than you expected. And these guys as positive because it will be much higher. So as you can imagine, obtaining this information is very valuable for understanding what the relationship between genes. And more than a decade-long project in Toronto is aiming at constructing and analyzing all possible combinations of double mutants in a single model organism, which is yeast. And yeast has 6,000 genes, so there's a total of 36 million combinations. And I'm going to talk about today some of the work we've done up to a few years ago, and also, if I have time, a little update on where we are now. And the short version is that the project is almost complete. So once we and others have started collecting this data on a systematic scale, on a large scale, people realized that not only individual interactions were informative, but also knowing that two genes share many interactions in common of the same type was extremely informative about their relationships. We found out that members of the same protein complex, for example, will have a very high correlation between their genetic interaction profiles that are defined just as a set of interactions between a gene and all the other genes in the genome. Of course, with data like this, clustering, large matrices and clustering have been applied very early on. But once the data starts growing, clustering becomes very, very impractical, both because it's just a huge matrix to deal with, and also because it becomes more and more difficult to find interesting trends in this giant matrix. So one approach that we have borrowed actually from a neighboring field is to represent the same data as networks. So basically, what you're doing is you're taking all of the data from this data set, computing pairwise correlations between every pair of genes that you have, and representing them as a network where nodes are genes and two genes are connected by an edge that's weighted based on the correlation of their profiles. This is very similar to co-expression networks, for example. The only difference is what data goes into the correlation. And then you can take this network and apply a network layout to it, that I'm going to get back to in a second. In particular, a class of network layouts that are called force-directed that will reorganize the network in two-dimensional space such that tightly connected nodes will be close to each other in 2D, and the guys that are not so tightly connected will be free to move and eventually will be pushed away. At the end of the day, so network layouts have been around for quite some time, but surprisingly, they've been used mostly for presenting networks, creating visualizations. However, if you think about it, the correlation part and the layout part are effectively multidimensionality reductions approaches, right? You're taking a gene from a very high dimensional space and placing it on a 2D space relative to all the other genes in the genome or in your data set in a way that makes sense, that it's completely data-driven. So when we applied this idea to the entire network back in 2010, that's the network we got. Once again, genes here, a node are represent genes, and two genes are connected by an edge if their profiles, genetic interaction profiles, have a Pearson correlation greater than 0.2, where both Pearson correlation and 0.2 are completely arbitrary choices. So I will get back to that if I have time. So obviously, the first time we saw this we thought it was very interesting because, as you can see, genes are not randomly distributed throughout this network. They form these very large groups or clusters, if you can want to call them, that are very densely connected and are clearly much bigger than complexes or pathways that we knew or that we expected from this data. So we went in a lot of trouble trying to annotate this network. Yeah. So there was one giant connected component, which is this one, and probably maybe 20 more disconnected, which probably are driven by the applied cutoff. Yeah. You can dial it up or down depending on your computational power, how much your computer can handle in terms of representation. Yeah. That's what, yeah, that's what the 0.2 comes from. That's at the time, that's what my computer could handle. But of course, I mean, it's definitely the way to go is to make every analysis independent from that cutoff, or as independent as possible. Yeah. And actually, so I have data suggesting that the analysis that I'm going to present is fairly insensitive to the specifics of the cutoff within a certain range. Okay, so we went into a lot of trouble trying to understand what the giant clusters on this network represent, and we did a very complex analysis based on some clustering, but mostly manual investigation of the network. In other words, we zoomed in particular regions, look at the genes, and try to assign a label to them that made sense based on what we knew about the genes. So it's a very kind of thorough but extremely long and time-consuming procedure that took about probably several weeks to accomplish. Well, the good news that when we finished was that we could assign a label, and we're very confident about the functional association of every region of the network with a particular biological function. So for example, this region was clearly enriched for secretion and vesicle-mediated transport genes. This region over there was clearly DNA replication and repair, right? So that was roughly what we expected, but at the same time was interesting to see that it's actually true. So since then, obviously, so this has been 6 years ago, the Toronto team has kept screening, obviously, and produced a large amount of data that unfortunately I don't have too much time to go into, but the end point of that is that from a network this big in 2010. In 2016, we had a network that was largely 50 times larger, and in reality was composed from 2 independent networks derived independently experimentally. One that was composed entirely of non-essential genes, and the other one that was entirely essential. And then the combined network is the one that was much bigger, corresponding to the one we had before. So because we basically obtained 3 new networks, and all of them were larger than the original that we started from, when the question was asked, okay, so what are these networks? What's new about them with respect to the old one? It was completely unfeasible to redo the same analysis we did last time, because it would just take forever and would not be reproducible and just not the way to do it. So what became apparent is that we need an automated and unbiased way to perform the same operation, basically to answer the question of what is what on this network, what functions are represented, where are they, how they're related to the previous version of the network, how they're related in the 2 different parts of this network, to answer all these questions in an unbiased and systematic and hopefully quantitative way. And so that was part of my job as a member of the team, and what I came up with is a very simple idea that's actually builds on many of the discoveries and research of many people in this room, but it worked extremely well for the kind of questions we had. And this method that we came up with, we call it Spatial Analysis of Functional Enrichment, or SAFE for short. It's based on the idea that you can take any gene, any node in that network, and define a local neighborhood around it. Local neighborhood, all it means is a set of the other nodes that are located closer than a certain distance. So of course, as we heard earlier today as well, there's many different ways to measure distance, both shortest path length or some maybe I don't know, decay functions that can, exponential decay function that can be used to give different weights to nodes as you move further and further from them. But one option that we have considered was to take actually advantage of the layout algorithm that we have employed to make the network. And we measured distance between nodes based on the physical length of their edges once the layout has been applied. So in this case, all genes, all the nodes B labeled here are part of A's neighborhood, because the sum of the edges that you have to cross to get from A to the node is less than a certain threshold that we have chosen. Once we have defined the neighborhood around A, we can run simple functional enrichment on it. For example, we can run a hypergeometric test against any given Go term. Take the p-value that is derived from that analysis and assign it back to the node we started from. Yep?
So in order to be able to apply this, you have to have a rough idea that your method is robust, meaning one run after another gives roughly the same distance.
Yes. Yes, yeah. We validated, so we did it in reverse. We first applied it on the one network we had. Once we got the result, we went back and made sure it was robust to many independent runs. Yeah.
And the second question is, compared to the 2010 network, 2016 network preserves the general features, similar genes appear in the same place in the network. Is there robustness with respect to insertion of more nodes and more edges?
So I don't have a number to tell. Obviously, if you add more data, some things will going to change. But they will show at the end that even, I would say that roughly nothing really major changes. I'll get back to this. It's easier when I explain the process. Okay, so as I said, so you take the p-value for any given Go term, assign it back to the original node, and then move on to the next node. And you repeat this operation for every node in the network. And at the end, what you have is a set of p-values assigned to every node in the network that indicates how much that node's neighborhood is enriched for a given Go term. So I've done this for all 4,000 Go terms associated with at least one yeast gene. And what I found is that there's quite a difference in how Go terms behave across this network. There's about 12% of Go terms that look like this. So here, I'm representing, this is the same network, so it's basically a representation of the same network, except that I'm not showing the edges, but the node positions are exactly the same. And each node has an opacity that's proportional to the negative log 10 of the p-value for this particular Go term. So what it says is that this region and this region only is enriched for ER to Golgi vesicle-mediated transport. And there is no other region on the network that with these parameters shows that same enrichment. And 12% of Go terms behave like this. There is another 4% of Go terms that behave similarly, but now they're enriched in multiple places. We call them multi-regional. There is a relationship between these guys and these guys in the sense that these tend to be larger in the number of genes annotated to them. But it's not a very clear distinction. So size is not the only thing that drives this difference. There's many very large Go terms that look like this, and many fairly small Go terms that look like that. That is an interesting feature that, if I have time, I can go back and discuss. And then I guess the most interesting thing was that actually the vast majority of Go terms didn't show any signal at all. So they were either too small to be represented on this network, or they are represented, but they just don't concentrate in any particular way. I'm not trying to say that these Go terms are bad. I'm just saying, in my interpretation, this means that these Go terms do not agree with this network. That means that the way these Go terms group genes is not the same way the network groups genes. Yeah.
There's also a biological explanation, because you are asking for an effect, a genetic effect on cell growth, right?
Yeah. Mm-hmm.
And if you have terms that do not affect directly the cell growth,
yeah. Absolutely. So it's completely network driven. Whatever the network is, if the network doesn't capture certain phenotype, like certain biological processes, they will not be represented here. It's completely network driven. Okay, but so the good news is that if I take these 12% of Go terms all together, cover all of the multi-regional terms. So there is at least one Go term that is represented in a region that it's enriched for at least one multi-regional terms. So for the rest of the analysis, I'm going to ignore these guys altogether. I'm only going to focus on this 12%, which is about 500 Go terms. The other thing to consider, obviously, is that Go terms are not perfectly separate from each other. There's a huge amount of redundancy, especially parent and child, right? So we can measure that redundancy in terms of similarity of their enrichment landscapes, which is this set of enrichment that you see in the network. So this guy and this guy, even if they're not identical, but they're enriched in the same region, and we can measure that. This guy is different from this guy because obviously they're enriched in different regions. So what we can do to try to summarize this all together, and so that you don't have to look at 500 of these plots, is we can assign the same color to these two because they're similar, but different colors to these two, and then we can sum them all up, basically compile them together into a single map that will just, for every node, will have a color that is the sum of all the other colors that is proportional to their relative enrichment. So if a node is more enriched a node can be enriched for more than one, actually. Most nodes are enriched for more than one Go term, but they might be enriched more for one than the other. So that Go term's color will be prevalent. So that's when we do this, we can create a combined enrichment landscape. And this is what it looks like when you do it on this real network. So here, so this again, it's the same network as that. I'm not showing the edges, but the node position is perfectly preserved. Every color corresponds to a group of Go terms enriched in that region. And there's about 19 regions here. And going from here to here takes about 20 seconds compared to weeks that we used to do before. Obviously, the one part that machine is not very good at is actually telling you what the regions correspond to. Obviously, every region has a long list of Go terms associated with it. But if you just want to glance, turns out if you just grab if you take the Go terms names and chop them into words, and then take the 5 words that come up more often, that's good enough. Right? It's pretty rough, but let's say this group is the 5 words are transport membrane protein Golgi localization. It's not perfect, but it's good enough to tell you what's going on. Okay, so just in the interest of time, I don't have a lot of space to show you the validation we've done, but we know that it's very accurate. It identifies all the regions we identified in the manual analysis, plus 3 more, tiny little regions, so not surprising we missed them before, but still, it shows you that we can be more thorough now. It is robust to numerous sources of variation, but most importantly, which to me is the most important feature, is that it's fast and it's completely automated. There's very little human intervention there. And that means that you can take the same network and annotate it with many different independent functional standards, not just Go, because we know Go is not far from perfect in many situations. And the first thing that came to mind is that yeast, unlike any other organism on Earth, has been studied in many, many different contexts through thousands of phenotypic screens. The deletion collection has been subjected to many different stresses, and many different phenotypes have been measured. In particular, many chemical genomic screens have been done. And the beauty of chemical genomics is that they can help you understand what the target of a particular drug is by looking at what mutants in yeast show particular sensitivity. So drugs, in the oversimplification of the process, sometimes can mimic the phenotypes of their mutated target, which means that if you have a chemical compound and you know which genes show particular sensitivity to it, you can guess what the chemical compound targets because these genes are likely to be synthetic lethal with the drug's target. So that is very useful when you don't know what the drug does, because you can look at these genes, try to come up with an idea what their function they're associated with, and then try to guess what the drug does. So as a test case, and to test this with SAFE, I took one of the most recent chemical genomics data sets in yeast that has, as a subset of their data, 132 chemical genomic screens for drug with known modes of action. So there are no surprises there. I knew exactly what to expect. So SAFE had to be modified a little bit. So now instead of just counting how many neighbors belong to a Go term, we have their quantitative values associated, and we can sum them up and then run let's say 1,000 over 10,000 network randomizations and ask how often does this neighborhood get this score this high or this low in a random situation, and then get an empirical p-value out of that. So here's a few examples how that works. So this is the Go-based annotation just for reference. This is the same, the outlines are only shown for the regions that correspond to Go terms. And this is a profile for doxorubicin, which is a DNA intercalator that blocks replication. And in fact, the SAFE shows that only one region, the region associated with DNA replication and repair, is specifically enriched for sensitive mutants. This works only also when the drug has more than one target, as is the case for verrucarin A. It has one important function: it inhibits protein synthesis, and in fact we see that the ribosome cluster is particularly sensitive. But it has another second described function that is it's somehow toxic to mitochondria. It's not very clear why, but there's a paper in the 80s. I first thought it was a novel finding, but then I found the paper. It shows a toxicity to mitochondria. And in fact, SAFE shows that the region enriched for mitochondrial function is also sensitive. But the cool thing is that obviously, even within the known drugs, you can find novel biology as well. So here's a typical case of things you can find with this method. So bortezomib is a known chemical compound. Actually, is FDA approved it's an anti-cancer drug. And the safe profile showed that three major regions were enriched for sensitivity: transcriptional regulation, assembly of the proteasome, obviously, and cell cycle regulation. So bortezomib is a proteasome inhibitor, so this makes sense. It is synergistic with HDAC inhibitors, so this makes sense. And it is, people think that it's an anti-cancer drug because it prevents the degradation of pro-apoptotic factors. So in a way, it promotes programmed cell death. So probably the cell cycle regulation also makes sense. What didn't make sense is this thing over here. So this says that the Golgi, the vesicle-mediated transport function, actually mediates resistance to the drug. And as far as I could think or search through the literature, I could not find any indication of why that would happen. Nobody ever described anything like this. So the only link that I could find is that maybe by inactivating the proteasome, it increases the accumulation of misfolded proteins in the cell, and they are located in the ER, and maybe that causes ER stress. But why would mutating an ER gene, for example, make it better? I don't know. So that was a mystery to me. The only thing that I could think of is, what if this actually has nothing to do with the action of bortezomib? What if it is a side effect, right? It's just a chemical treatment. Who knows? It's an off-target effect. So the most direct way to test this was to say, well, if this is what I think is happening, so I have bortezomib inhibiting the proteasome, and and that blocks a certain pathway. But then when I mutate vesicle-mediated transport, I unblock another pathway and make it better. Then I should be able to recapitulate this exactly by not treating the cell with bortezomib, but by mutating the proteasome directly, right? I can create a double mutant in the proteasome gene and the vesicle-mediated genes, and this should be better than either one of them individually. So that should be a positive interaction. I was lucky because I actually had all that data already. I didn't have to do any experiments. And what I did is I took the genetic interaction data for many members of the proteasome, in particular this one guy, which is RPT4, is a member of the ADPase, an ADPase that's a member of a proteasome. And I found that its enrichment profile is remarkably similar to the bortezomib. So that means including this part, right? So this confirms that this thing that we see over here is a direct effect of the bortezomib inhibiting the proteasome, because I can recapitulate it exactly using a mutation. So just to summarize, what I've shown you is that we have a method that it's automated and systematic for annotating biological network. It answers, as of today, three major questions. Are any regions of the network enriched or specifically associated with a particular function or a phenotype? Where in the network this region is, and how does this region compare to other regions or other functions? It builds a functional map of the network and allows you to integrate multiple data sets together. I think I don't have any more time to go through any. Is that okay? Yeah. So I will skip to the acknowledgement. The rest was just to show how we have applied this to the new network. So this work was actually everything except for the experimental data was done in Princeton in my lab. Of course, many people, undergrads and technicians, to thank. The University of Toronto, of course, is providing all the data. We have a long-term collaboration with Chad Myers in Minnesota. And I should mention everything I told you so far. There is a manuscript on bioRxiv that describes all of it. There is all of the code written in MATLAB is on Bitbucket. There's a link from the manuscript. And we also put together a macOS standalone application that doesn't require MATLAB. And we're developing a Cytoscape app, which I think is cool. Anyway, that's it. Thank you. Yeah.
Give us maybe a bit of intuition how you deal with multiple hypothesis testing here.
Yes.
You have it in the Go terms because they're nested, but also in the network because the neighbors, of course, will have highly correlated significance.
Yes. So the way we so this is could be a complex question we could discuss offline, but the short answer is that we deal with multiple testing across Go terms. because every neighborhood is tested, of course, for 4,000 Go terms. So that p-value is corrected. We're very stringent. We use the Bonferroni correction, right? So every p-value is divided by 4,000, right, or multiplied actually by 4,000. In terms of multiple testing across the nodes, we do at least one thing, which is we filter by how many neighborhoods are enriched for the same Go term. But whether or not that actually addresses the concern of multiple testing across the nodes because they overlap, I would be happy to discuss that. In my thinking, it should address it, but I might have missed something. Yeah.
So have you applied that method to other strain of yeast, like WBC or?
Well, I can apply it to any network. So if the network exists, then yes, right? If the network doesn't exist, then I can do it. The genetic interaction network exists only for one strain, right? But I skipped this, but I did apply it to a protein-protein interaction network, and it does work, yeah. Yeah. Oh, yeah, Martin.
Do you treat all the Go terms in the same way, or do you have a difference depending on the hierarchy or the?
Yeah, no, they're absolutely completely agnostic to the size and the hierarchy of the Go term. So they're all it's a bag of terms, right, when we start. What we did do is once we have seen the difference between region-specific, multi-regional, and sparse, we see how they behave in the hierarchy. The thing is, there's no real correlation. No, so you would think that the parent would be multi-regional, and in some case there is, right? In the case that I showed you, protein localization is multi-regional, and localization to the R is region-specific, right? It makes sense. But there are many other examples. There's one famous one with mitochondria, where we see I guess it's the reverse. So we see all mitochondrial Go terms they are region specific. They're all in the same region. But if you look at Go, their common ancestor is the root, right? They're not closer to each other than basically any random Go term. And I think that reflects just how Go was constructed. These mitochondrial terms are biogenesis of mitochondria, energy production, and transport to mitochondria. According to Go, they're as far as they can get. According to our data, they're very close. So it can go both ways, but it's a very interesting thing. Yep. Sorry.
Yeah. So I get the rationale for visualization to do a 2D embedding of your network, but is there another reason why an embedding rather than just working with the graph directly, and why 2 dimensions?
Yeah, I mean, this is an interesting question. It goes, so I don't know, where should I start? So we started this from the idea like we wanted to look at the data. So it was motivated by visualization. It evolved into a more rigorous method today because we realized how powerful that could be, right? If you take into account all the subjectivity of visualization, right? For example, if the method is quantitative, if you have multiple testing correction, it does become a true rigorous method. And the embedding, as I said at the beginning, is not just for visualization. It could be used for real data analysis, right? It's driven by the data. The visualization reflects the topology of the network if handled appropriately. So I think my personal opinion is that this can be a very powerful, rigorous method, and it's not just for visualization. But the visualization adds an aspect to it that means that I can talk to biologists and they will understand what I'm saying. Anyway, sorry for the, yeah, oh, there's another question. Yeah.
So there are dozens of embedding algorithms.
Yes, yeah.
How did you choose?
So this one was basically what was available in Cytoscape. We tried a bunch, and this was the best. We are working right now, now that we've realized that this could be this powerful, we are actually developing our own algorithms that will take all of the information we think should be included. But yeah, definitely. There's a bunch of already existing, and we can develop more. Yeah.
I think we'll have to take the rest of these offline, but thank you again.
Thank you.